Diane, about what you were saying, I wonder, the convoys came out with a paper basically claiming that the spread of the data was more important than the traditional epigenetic measurements. And I wonder if that applies to what you were saying there.
Convoys also think if you continue to take out bad stuff, that you'll defy the laws of physics.
Well, I'd like that.
So would we all, although I don't know that I want to live forever. I just like to live healthy. But
laws of physics.
Yeah, because they're saying if you just keep replacing, if you keep taking bad stuff out, that your cells can continue to rejuvenate, and that defies the second law of thermodynamics, right?
Yeah,
It just,
well, especially if we don't know what that implies, which we don't.
They're just saying, again, keep taking out the old stuff and your cells will work. That you don't have to add anything else. And if you study, right, the laws of thermodynamics, there's a degeneration of the system based on math and physics. So anyway, I'm going to let Josh talk and kind of see what comes out.
Today's star has arrived, and by golly, it's almost 8 o'clock. Steve, oh, good. Hi, Josh.
Hi, Walter. How are we doing?
Oh, my. People are entering the room like crazy. Wow. Hey, we're gonna have a full house here from the looks of it. Whew. Good. This is great. Well, it's just such a wonderful subject. I would think that people would be pounding on the doors. It's so, I don't know the French, but it's very current.
Au courant.
That's what I thought, but I didn't want to risk saying it stupidly.
A la mode.
Yeah. John. Oh, John Ferber. This is great. This is like a regular rogues gallery of longevicists here.
Really?
Well, let's just give everybody, we're actually recording in case you don't know. So we'll give everybody, we'll allow them to be a little bit late. It's only 8 o'clock. Ooh, 18 people. Beautiful.
Hi, Walt.
Hi. Who said that? Oh, John. Oh, my old buddy, John. If it wasn't for John, I wouldn't even be in this room. Albert, you're upside down again.
I am a bat hanging from the ceiling. By the way, do we all know why bats harbor viruses that kill other mammals?
No, I don't. Everybody else does.
I don't know.
I'm always open to ideas. I don't have the answer either.
The answer to me is that the bat viruses have to be engineered to infect humans. And there's a profitable industry doing that.
Who's paying them?
Tony
Listen, maybe
Fauci.
it's the person who's hanging my picture upside down, gremlins in my laptop. I found out this morning if we were in WebEx, I'd be right side up. Apologize. I'll turn it off because it's a little disconcerting for some.
Oh, it's fun.
Well, I have to thank you for what looks like a new Canadian flag as a Canadian citizen.
Well, actually, it's half American, half Canadian, because my parents were Canadian. But you can't see the American half.
What did the old Canadian flag look like?
It had the little British overtone.
That was a long time ago.
I meant
was
new
time
in the
ago.
sense that it's freshly creased. So I'm thinking you recently bought it and put it up.
You know, it's been hanging there for 6 months. The creases don't go away. I need to come run some steam over it or something.
What's the blue and white cross behind you? I don't recognize.
Oh, that's St. Andrew's Cross or some Scots thing. Okay, here we are at 8:04. Do you want to get going? We have 21 guests. Oh, and more. Oh, my.
Looks like half of them are AIs.
So I am going to make it so that speaker gets the thing. And there's Josh. Go, Josh, go. It's 5 after. Oh, Darina, yay. Okay.
Is that what I'm supposed to be talking about? Is today's the day I'm talking about UFOs? No, okay. Exosomes. Okay, got it. Thanks, Walt, for putting this together. You've been my buddy for 20 years in research and in real life, and such a wonderful community of friends that you're connecting me with. Some of whom I've known for a long time. Some of whom are new to me through you. I want to talk about exosomes. I think exosomes are the most promising anti-aging technology that we have in the pipeline now. And it's based on the foundational work of Harold Katcher, my friend and colleague, and corroborated just this spring by a study out of Nanjing University demonstrating robust rejuvenation by many different measures. I do not think that exosome technology is ready for the clinic yet, but it should be a high priority. And that's really what I'm here to convince you, that translating exosomes from rats to humans is the most important work we can do in the anti-aging field right now. I'll describe some of what I think I know about the research, and I'll frame it all in terms of my own paradigm of what aging is and how aging works. But first, you get a taste of my philosophy. No apologies. Reductionism has become synonymous with science in the last century or two. And what is reductionism? It's starting with the smallest things and building them up, understanding each level in terms of the properties of the things before it. Emergent is the operative word. Structures at the larger scale just happen as a non-obvious consequence of a lot of small, simple systems interacting. And sometimes this is exactly what is going on. Snowflakes form these wonderfully hexasymmetric crystals, and there's nobody imposing a mold on them, making them this way. They really are emergent properties. Traffic jams emerge from individual behaviors, and there's nobody guiding the traffic jam and making it happen. But it's not always true that behaviors of large systems are emergent. For example, we have this dogma that says that the greatest good for the greatest number will come out of wildcat capitalism. All you need is free markets, and free markets don't necessarily lead to everybody's welfare. The invisible hand that works in capitalism sometimes makes misery for everybody. And there's no guarantee that selfish genes are not going to create competent individuals, let alone balanced ecosystems. So trying to emulate the success in physics, biology has become very one-sided recently. The whole science of biology has shifted toward biochemistry since the mid-20th century. The mainstream of evolutionary scientists have been trying to explain everything in nature based on selfish genes, and system biology has suffered. And on yet larger scale, ecology remains understudied and underappreciated. Ironically, physicists have gone in the opposite directions because quantum mechanics is inherently a many particle system, and you've heard the word entanglement. The concept of entanglement is baked into quantum mechanics at a deep level. You've probably read about the two particles that interact and then they fly apart and they remain connected to each other. Well, that's just the simplest example, and it's one that can be calculated. But in fact, everything is related and entangled with everything else. If you think this sounds like Oriental mysticism, you're not the first person to say so. Schrödinger and Bohr and Planck were all interested in ancient Hindu scriptures, not to mention Pauli, who was a genuine mystic, and Bohm, who had relationship with Krishnamurti. If you have a chunk of metal with 10 to the 23rd electrons in it, it doesn't have 10 to the 23rd wave functions. It has one wave function that describes the configuration of all the electrons. The wave function must allow explicitly for the fact that these electrons are identical, and therefore you don't know which one is which. It's much stronger than what you think of identically, two identical ball bearings. In fact, built into the fundamental equations of quantum field theory is the possibility that whenever we're not looking, electrons can change place with each other, like Eddie Murphy movie or Mark Twain's Prince and the Pauper, one electron becomes the other electron. And this is explicitly written into the probabilities of what's happening in quantum field theory. Yes, physicists do still think in terms of individual particles, but it's not because this is what their equations say, it's because the equations are so damn complicated that once you get past two particles, you can't solve the equations, even with a huge supercomputer. And so physicists calculate one particle wave functions because that's where we have solutions to the equations. Meanwhile, biology has become grossly unbalanced. We know a great deal about molecules. We know something about cells. We have a rudimentary knowledge of what organs and tissues do. We know very little about whole organisms and how they work as a system, and even less about ecological relationships. My work on aging began 28 years ago. I began with the paradox that aging has evolved despite the fact that aging is the opposite of fitness, which is supposed to be what evolution is aiming to optimize. I spun my wheels for the first 5 years trying to understand, where does aging come from? And I only began to see an answer when I realized that aging has a function that transcends the individual who ages and dies. Aging is an agent for stabilizing ecosystems. You can't have life without ecosystems, and you can't build ecosystems out of selfish individuals that are trying to live as long as possible and to leave as many offspring as possible. So this is my principal contribution to the science of the evolution of aging, to realize that it all derives from the fact that we need ecosystems and that ecosystems are impossible without birth control and death control baked into the genes. Today, there are still people who are trying to understand aging from the level of cells wearing out and molecules that suffer damage. But 20 years ago, the Conboys and Amy Wager, while they were still working in Tom Rando's Stanford lab, demonstrated that there are signals in the blood that control aging, not at the cellular level, but at the system level. Today, the only successful anti-aging interventions that we have are modifications of the signaling environment. Researchers who are trying to repair damage at the cellular level and below are not getting anywhere. But they're diverting a lot of our resources and some of our best scientists into what is patentable instead of what works in aging. I'm not one to celebrate the huge influx of venture capital that has come into the aging community in the last decade. I think it's great to have more money, but where the money is being spent is distorting the field. I've written a series of papers on the question, how does the body know how old it is? I don't think the system just wears out. I think that our bodies are actively destroying themselves, and at the same time, slowing down the repair and detox processes that are perfectly adequate when we're young. This implies that the body does know how old it is and acts accordingly. The body is a system. It's not individual cells acting independently. Aging is coordinated system-wide, and instructions are communicated to every cell for age-appropriate behavior. Is there some central clock, or is the clock dispersed somehow throughout the body? Maybe there are several different clocks. This is an unresolved question, something of an embarrassment to people like me who are proponents of programmed aging. But in my defense, I'll say that we don't know what time's puberty either, or the cessation of growth when the body reaches full size. And yet we're confident that there are such clocks. Nobody out there is arguing that the female body turns on sex hormones and starts to menstruate when a certain level of damage accumulates. No, we're confident that there is some kind of clock or clocks, even if we haven't discovered them yet. So my guess is that the same clocks that control puberty are extended to control aging as well. And if we could find that clock and reset it, like rolling back the odometer on a car, we could make ourselves young again. We can't do that yet, but we might be able to do the next best thing. The body does know how old it is, and that information has to be communicated, dispersed to every cell in the body. Starting in 2005, I mentioned the Conboys and their other students of Tom Rando, who showed that there's something in the blood that communicates information about age through the body. The subsequent work by the same people demonstrated that it wasn't the blood cells, red or white, so it must be something in the plasma. And starting around 2018, Harold Katcher made an inspired guess about what that something is. His guess was exosomes, but he didn't tell us that. We don't know how the body keeps track of age. We do have a good candidate for how age information is transmitted through the body, and that is exosomes. Here's my current hypothesis. Exosomes are flowing through the bloodstream all the time, excreted by every cell, picked up by other cells. They carry all kinds of information. Some of them carry age information. Different cells have different ideas about the age of the body, and exosomes are a negotiating tool. The body arrives at a consensus age by spreading information through exosomes. I don't think the process is entirely democratic. I think some exosome sources are more equal than others. It may be that exosomes from the hypothalamus in particular are more plentiful or more influential when it comes to negotiating the age state of the body. So taking a step back, what is an exosome? The word extracellular vesicles appeared, I think, 30 years ago, 40 years ago. These are packets of DNA, RNA, proteins wrapped in a lipid nanoparticle and secreted by just about every kind of cell. They're categorized crudely by size because we know so little about them to categorize them better. Their size ranges from 30 to 100 nanometers. Those in that size range are called exosomes. They're much smaller than bacteria, which are in turn much smaller than the cells in our body. They're about the size of viruses. Up until about 20 years ago, the dominant theory was they're just excretions of molecules that the cell doesn't need anymore. They're waste disposal. Exosomes were thought to be waste because coordination at a higher level was something that the biology community dismissed. But then we learned that these exosomes are picked up by other cells. Then we learned that they carry information that affects those other cells. And then we learned that the information that they carry can cross species lines. For example, warning other animals of an invading pathogen or warning other plants of an invading insect. Then we learned that, like cells, exosomes display protein markers on their surface. In the case of exosomes, these act as address tags that help determine which cells will pick up the exosomes. So as best we can tell, exosomes constitute a universal signaling language, very complex. We've just begun to plumb the kind of information that's communicated, not only within our bodies, but across species as well. Trees in the forest shed exosomes, which alert other trees that there might be a certain fungus infecting them or some insect in the neighborhood. If you're infected with a virus or a bacteria or a parasite, you will exhale exosomes with pieces of the genome from that infectious agent, and I will inhale those exosomes, and my immune system will learn from your immune system and be better prepared when the infection comes my way. And of course, I'll pass on that information to others. Incidentally, this is one of the reasons that it was a really bad idea to lock everybody down when COVID was starting to circulate. Lockdown slowed the spread of virus in the short run, but in the long run, everybody got exposed to the virus anyway, and their bodies had less advanced warning via exosomes to prepare their immune systems. Lockdowns made the pandemic unfold more slowly, but ultimately, I believe, killed more people as a result. So, sideline here, the evolution of viruses. It's a plausible theory that viruses came from exosomes. Sometimes the message that comes in an exosome is deemed by a cell so important that the cell will copy the exosome and send copies of the exosome out to transmit more of that information. So you can imagine that this process is hijacked by a selfish exosome that doesn't have any message to carry, but just reproduces itself, hacking the system. This is a plausible story for how viruses evolved. So what is the evidence that exosomes have rejuvenating power? I mentioned the Stanford lab of Tom Rando, where they started, Mike and Irina Conboy came up with this protocol of tying two mice together, sewing them together with denuded skin in the interface. The mice grow together and share one circulatory system. If you do this with an old mouse and a young mouse, they found that the old mouse shows signs of rejuvenation and the young mouse showed signs of accelerated aging. The Conboys set up a lab at Berkeley where they demonstrated that it wasn't blood cells, but something in the plasma responsible for the effect. Amy Wagers was doing comparable parallel work at Harvard. The Conboys are now working on removing the old signals from blood, and they're working in cooperation with Dr. Dobry Kiprov in the Bay Area. There's an ongoing human trial simply removing old blood plasma, not the red or white blood cells, as a therapy. This is called plasmapheresis, and it's recognized and sanctioned by the FDA. It's grandfathered in because it's an appropriate therapy for some autoimmune disorders. So we don't need to worry about regulatory approval. But what about getting young exosomes into the body? I first heard about this from James Watson, the other James Watson, not the DNA James Watson. He told me about exosomes, but I was too distracted to listen to him way back then. But 7 years ago, Harold Katcher made an inspired guess about what it is in the blood that's carrying age information. And he set up shop in Mumbai, funded by Akshay, infusing old rats with blood plasma from young rats. And he called it a plasma fraction in his article. He called it elixir or E5, but wouldn't tell us what it was. In 2020, his team announced that they had turned 2-year-old rats into 1-year-old rats, according to the Horvath multi-species clock, which was developed really just for that purpose. And I wrote about this in my blog as a breakthrough in age reduction. But then from 2020 to 2023, research proceeded very slowly because Akshay had trouble raising funds for industrial scale extraction of E5, and nobody else knew what E5 was. But then last summer, a research group at Smith Heart Institute in Los Angeles announced promising results rejuvenating the hearts of old rats with exosomes from young rats. And the hearts functioned better, and there were fringe benefits for other organs as well. Exercise endurance improved. They didn't do a full lifespan assay, but they did notice that the treated animals were dying at a lower rate than the untreated animals. For experimental controls, they treated old rats with blood plasma from young rats, but from which the exosomes and nothing else had been removed. And that young plasma had no benefit. So this constituted really the first strong evidence that the exosomes are the ingredient in young blood that has rejuvenating power. So Akshay had been trying up until that time to keep E5 a secret, but now that others were pushing the idea, Akshay sent me this article, in fact, and his hand was forced. He was put up or shut up, and he consented to reveal that the secret of E5 was exosomes. E5 was exosomes. The next question is, would the phenotypic and epigenomic rejuvenation that Harold and Akshay observed and the Smith scientists observed, would it lead to dramatically longer lifespans? We have data from only 8 rats. They're all female. And those results are not as consistent as we might hope. The median lifespan increased about 20%, depending on what you use for baseline. But curiously, the maximum lifespan seemed to be extended by 60% or more. This past winter, you may know that Akshay and Harold parted company. They're pursuing separate paths toward commercialization of exosome technology. And then just this spring, there was another research article from Nanjing University in China. It added many more physiological and behavioral measures to Harold's data. They didn't report Horvath clocks, but they did work with male rats this time, and we have lifespan data that you see shows a greater increase in maximal lifespan than in average lifespan. So there were many measures of rejuvenation that they found in this study. Exosome-treated mice scored much lower on a frailty scale than untreated mice, not quite fully restored to the zero frailty state of youth. Sperm counts were restored to young levels completely. Testosterone levels increased. Treated mice were as fertile as the young mice. Metabolic rates were measured by oxygen consumption, also restored about halfway to youthful values. And the activity of the mice increased in conjunction with that. Ejection fraction and other measures of heart performance were improved, but not to the level of young mice. Parts of the brain that atrophy in old mice regrew toward youthful volume. Memory was measured by a water maze test and restored to youthful levels in the exosome-treated mice. Treadmill endurance came back almost to youthful levels. The treated mice had lower markers for the molecular linkages that link sugars together and that increase in old age. Glycation. So glycation decreased in treated mice, but not quite to the young levels. Treated mice were restored from osteoporosis to higher bone density. They did not report results from the Horvath clock. However, they did their own analysis of protein expression in various tissues, and they showed that proteomes of treated mice reverted statistically in the direction of younger profiles. They used as a control young mice infused with exosomes from old mice, and both the endurance and the memories were impaired. So the exosomes in old mice have the power to make you old, as well as the exosomes in young mice make the older mice young again. I'm not the only one who's noticed this.
Josh?
Yeah.
Pardon me. If you could just take a moment, I should have done a little bit up front to talk about format and so on, but is there anything in the messages that needs to be addressed before we get too far along?
You want to take a break and answer questions?
Well, that wouldn't be a terrible idea. John Hemmings is an amazing fellow, has a couple of interesting points.
Yeah, I'm not moderating the chat, so I'm counting on you.
All right, here we go. He says, well, can you see the chat?
No.
Okay, you can actually. You just hit chat on your lower bar.
I can't and still have the, okay. I don't know how to do that in the PowerPoint going.
John, go ahead and say what's your
please.
Points.
Yeah, it's the point about exosomes. This comes back to basically that the mechanistic theory as to why aging occurs. And I start out with an assumption that it's a variation in gene driven in part by the mitochondrial membrane potential. Now, exosomes are in part used to distribute mitochondria around the body. Not the only thing they do, but it's a very important thing. If you look at Matrix Bio's work, they're very good work. Matrix Bio do some excellent work on looking at how you can give people mitochondrial transplants and the like. And, you know, the fact that certain exosomes are used for transferring mitochondria is important. But also, to be fair, macrophages transfer mitochondria. And also, you've got interleukin-10 with its effect on senescence. So it's another factor as well. And so, yeah, I would argue the case that exosomes are important, but that they're important because they transfer mitochondria, which is the sort of HIF1 alpha stuff as well.
I didn't know that exosomes include mitochondria, and that sounds like a hypothesis that can be tested quite
It's
easily.
not a hypothesis. I can, if you want, well, tell you what, you keep going and I will rummage around and
You're saying it's already proven that the...
I'm pretty certain it's already proven.
Yeah, likewise, I actually was looking into it yesterday and found that, because I wanted to make sure, because I was a little confused by some things I'd read, but it looks like mitochondria are in toto
John,
carried through the system.
You were saying gene expression, that epigenetics is at least how aging happens.
Yes.
Do mitochondria outside the nucleus control gene expression within the nucleus?
Yes.
They do.
Yes.
So I'm open to learning about this.
None of this is new. This has been around for some time. Mitochondria, they export loads of things, not just ATP. Key two things are obviously alpha ketoglutarate or 2-oxoglutarate and citrate. Citrate is the key driver. Citrate, once within the cytosol, is converted to acetyl-CoA. Acetyl-CoA is used in the nucleus. It's used for lots of things, but it's used in the nucleus to acetylate the histone. And without acetylation of the histone, you don't get mRNA. And so it's a key driver in splicing. You know, you've got different splicing variants. And the key driver is levels of cytosolic acetyl-CoA. So none of this is massively new. It's been around for some time.
I'm open to learning from you.
Well,
I'll certainly read more about the role of mitochondria. If I had to guess, I'm sorry, we're talking at the same time because of the delay. Go ahead.
But it's difficult with these things. But I have my own hypothesis as to why aging works, which is linked to the levels of acetyl-CoA in the cytosol.
I want to learn more about that. If I had to bet right now, I'd bet that it's complicated and that all of the components of exosomes work together. That it's not going to be just one mRNA, one RNA species, or it's not going to be just mitochondria.
I think a lot of things will have an effect. The mitochondria are a key element.
How does the mitochondria, in your perspective, how do mitochondria know how old the body is?
Because it's about heteroplasmy.
Come again? I don't know what heteroplasmy is.
Okay, you got your mitochondria. You understand endosymbiosis, yeah?
I don't know how that relates to, how does the mitochondria keep track of the age of the body?
Through the mitochondrial DNA.
Is it changing with age?
Yes. Not only yes, hypothetically, but yes, evidentially. This is where Tom Benson, who runs Mitrix Bio, had done
maybe you'll give next month. Maybe you'll give next month talk. I don't mind. No, I've done about it
with various people. It's no problem, but it's a good discussion to have. And
I don't want to be dogmatic about it, and of course I'm open to changing my mind.
So
prepare to speak based on what I know at this point.
Let me intervene a bit. Rather than get completely sidetracked with this very interesting discussion,
switch off again.
It is really complicated, but also, Josh, before you go back to your presentation, we have John Ferber asked, were the mice that were sharing exosomes genetically identical? I guess that applies to both labs.
So the rejuvenation, not only weren't they from genetically identical mice, they were from pigs. Exosomes from young pigs were used to rejuvenate old rats.
But were the rats identical?
The rats are identical to each other, but the exosomes in Harold Catcher's work were exosomes from pigs, which is, it's very encouraging that they cross species barriers. John, does that answer your question, or were you?
Yes, that's very startling. And thank you for pointing that out.
And then Steve Beck-Buss also entered a comment saying Chen 2024 demonstrated, among other things, that mitochondria are impacted by exosomes.
So the exosomes carry information to mitochondria as well as carrying mitochondria through the body.
In that same article demonstrates in experiments at least one known target, younging promoting exosomes, is a target called PGC1A. Among other things, there are multiple known ways already that can profoundly increase PGC1A, that including hyperbaric oxygen therapy, and I would say EWA, exercise with oxygen therapy by inhalation, also PEMF, and also red light therapy, and increase.
Steve, for some reason you're fading a bit.
I'm sorry. So there are interventions that increase the molecular target.
I don't know what PGC1A is, and I don't have a context for its role in aging.
Chen, I thought there was a link to that study in Alter's.
I don't know what PGC1A is.
Specifically, in the abstract, they say that extracellular vesicles, and this is a quote from the abstract, reverse degenerative changes and age-related dysfunction, at least in part by stimulating PGC1A expression and enhancing mitochondrial energy metabolism. That article is not about any semantics.
I can no longer hear you.
You moved away.
That article is a masterful study because it isn't a single thing. It's about a group of experiments from high-level analysis down to looking at microRNA and this molecule PGC1A.
So when I wrote about that study in my blog a couple of months ago, it seemed to me that they had demonstrated the effect much more convincingly than they had demonstrated the mechanism. And I don't believe that they have nailed down the mechanism. For example, you take away one component and I think they took away 3 RNA components and they said, well, these 3 seem to be the payload that has the rejuvenation potential. And it doesn't prove that. All it proves is that these 3 are part of the recipe, but the recipe may be more complicated. I'm taking the position that we still need to do more basic research. And what I was hoping to say coming up is that we need to do basic research, not to discover the mechanisms, because the mechanisms probably will prove to be more complicated than we can handle, but that we should work with different sources and different kinds of exosomes and actually measure their rejuvenation potential, rather than trying to drill down deeper to specific molecules within the exosomes. And, I'm open to the other possibility that maybe it is just one or a couple of species within the exosome. Maybe it's the mitochondria within the exosome that are doing all the work, and we don't need the whole exosome, which would make things much simpler in our rejuvenation work. But I'm not betting on that at this point.
Now, John has added a couple of more comments here.
Yeah, please.
I think I would say just, and I'll stop, but quickly, Chen 2024 et al. Says that age-related dysfunction is reversed by stimulating PGC1A expression. HBOT, hyperbaric oxygen therapy, has been shown to increase PGC1A expression, and the experiments of Efrati in Israel around hyperbaric oxygen therapy demonstrate profound impacts on telomere length, senescent cells, and other things. That's an amazing.
So that my perspective on that study out of Israel was that it was overhyped. What they demonstrated was longer average telomere length. But if you look at the mechanism, it seems very possible that they were eliminating senescent cells, which had the shortest telomeres. And the reason that the average telomere length went up was that they were eliminating senescent cells, not that they were growing telomeres. Now, that's not a bad thing. If hyperbaric oxygen eliminates senescent cells, that in itself has rejuvenation potential. But I wasn't convinced that hyperbaric oxygen expresses telomerase.
You want me to comment on this?
Yeah.
Well, first thing is there's a lot of evidence in terms of oxygen therapies that it is the reduction in partial pressure of oxygen that stimulates HIF-1 alpha. And HIF-1 alpha is a key driver in lots of different things. So HBOT works not because of the high oxygen, but because the oxygen levels reduce. And that stimulates.
It's when you come out of the hyperbaric chamber.
As you come out, this is where the Israeli experiments used a 20-minute period and reduced the oxygen levels. I wrote about it on my blog some time ago. There's a lot of studies, what's called the normobaric oxygen paradox, which is you can take the partial pressure of oxygen, not the overall pressure of gas that drives everything. And it comes through to the mitochondria, but in this instance, it's HIF-1 alpha. So that's where your HBOT stuff comes from, because
has anybody tried doing the same thing without the hyperbaric oxygen?
Yes, it's
going
on my
to high. Excuse me. Has anybody tried without the hyperbaric oxygen, just going to lower oxygen concentration as breathing 90% nitrogen and 10% oxygen instead of 80 and 20?
In a sense, the naked mole rats do that all the time, and so do the bowhead whales. But there are experiments done without hyperbaric pressure. So in other words, with normobaric pressures.
But 100% oxygen.
Well, it's not 100%, but like 90% oxygen. This has been done plenty of times. It's not new stuff. It's been around for some time. That's your HIF-1 alpha saga, which is useful for improving the mitochondrial membrane potential. And the mitochondrial membrane potential drives, to some extent, the SLC25A1, citrate carrier in how much citrate comes out of the mitochondria.
Didn't I read that going back and forth between high oxygen levels and low oxygen, subnormal oxygen levels actually is therapeutic?
Yeah, that's what it is. It's actually, the odd thing about it is it's not the constant state. It's not the steady state that matters. It's the reduction in partial pressure of oxygen.
I've also read the converse, that the increase in partial pressure of oxygen is damaging.
Well, no, a high partial pressure of oxygen is damaging. That's not the delta, that's the actual static, because it creates more ROS. The difference is the metabolic response is actually from the change. It's not from it being a low level or high level.
Just to reiterate, I've heard that the change in the other direction
that's interesting.
is damaging.
I'd be interested in seeing your references. I wrote a blog post on this some time ago, which I can give you a reference to. Which
please.
Pretty well.
Send it along. You've given me lots of stuff to read, and I'll go home and do my homework.
I'll hunt that up. I'll go quiet for a bit, if you don't mind, whilst I find it and put the link in the chat.
John and Josh definitely have reason to get connected. Now, I see that Albert also has offered a little something. Can you summarize it, Albert?
Thank you, Walt. I think I just did in the chat, which is that in terms of activating mitochondrial activity and response to hyperoxygenation or hypo, there is a pathway that responds to both. It's called hemoxygenase 1, and it's one of the many heat shock proteins found in exosomes. The slide we saw before that showed the contents mentioned or showed HSP 70 and HSP 90 that respond to heat and other stressors. HO1 does as well. Its prior name was HSP32, and it's the only one of the three whose output includes carbon monoxide, bilirubin, and ferritin.
So let's get back to Josh's presentation because Johnny's waiting in the wings here. Go ahead, Josh.
So what's the timing here?
There's no specific timing.
All right, I'll just finish up what I have to say. So why am I so enthusiastic about exosomes, given that the life extension that we've seen so far is not extraordinary compared to some other interventions that we have? I'm enthusiastic because this is the first time that we've seen actual rejuvenation, setting back the Horvath clocks and these many markers of age that have been measured by the Nanjing group and by Catcher. The intervention didn't begin until the rats were 2 years old, which should have been the beginning of the end for your average Sprague-Dawley rat. Add to this the fact that we're in a very early stage of investigation. We haven't begun to optimize the timing or the dosage or the kinds of exosomes. In Catcher's experiments, rats were rejuvenated with exosomes from young pigs, and this is encouraging because it suggests that exosome rejuvenation crosses species barriers, so we might be able to mass produce a human therapy from the millions of pigs that are slaughtered every year for pork. There may be problems. Harold has indicated to me there are problems because that pig farms are such horrible conditions and the factory farms that the pigs just are not fit for producing healthy exosomes. We have to work that out. Other people have told me exosomes can be extracted from stem cell cultures, and human placentas and umbilical cord tissue are routinely discarded by hospitals and birthing centers. These may be another source of human exosomes. We haven't even tried this yet. I think there's a lot of laboratory work that can be done to optimize these results, and I'm hoping that we'll see greater rejuvenation when the technique is refined, when we combine removing old exosomes with adding young exosomes, and when we target the specific kinds of exosomes. Among the kinds of work that need to be done, we need an industrial scale extraction technique. These are all names that I don't really understand, but I've heard that these are techniques for extracting exosomes from blood, and they all work well at a laboratory level in creating tiny quantities of exosomes suitable for a mouse. We're going to need a lot more if we're going to do larger animals or humans. There may be a paradigm for investment, a challenge for the investment paradigm in here. Where do you find IP in this? It may be that, as John says, there is some molecular species and something that's patentable in here that's optimized. My assumption has been that the rejuvenating power comes from whole exosomes, and there are so many species in there that will be decades untangling the molecular mechanisms, and we want to use exosomes for medical rejuvenation long before that. And this brings me back to the reductionist paradigm. Western medicine, the pharmaceutical industry, has been looking for single molecules, and their model is, well, one molecule, one effect. If we were designing a body, sure, every part would have one function and it would be there for a purpose, but our bodies are not designed that way. Biology does not work the way a machine would work. Every molecule has many functions, and every function is a result of many different molecules. Makes you think of the traditional Chinese medicine pharmacies that took many herbs and compound them together. It's an art as well as a science. There may be 5 or 10 different herbs in a prescription, and of course, many thousands of different molecular species. TCM actually works, even though we don't know how those individual molecular components work together, which ones are more important, which ones are less important, how they interact. But in practice, we can use the recipes from TCM because they've been studied as a whole. You apply the exosomes that we have and you look at the rejuvenation effect, and we'll be able to make progress in the field without understanding the detailed molecular mechanisms. So here's the challenges that I see going forward. We do need to check that they cross species lines, that exosomes from animals are able to rejuvenate humans, or else we need to grow exosomes from human placental or umbilical tissue, or perhaps from breast milk, which has exosomes. Do these exosomes from stem cells and from breast milk, do they have rejuvenation power? Nobody's looked at that as far as I know. This is experiments ripe to be done. I mentioned that we need to scale up exosome extraction. Exosomes come in different types, not just by size, which is the only way in which they've been differentiated so far, but they have protein markers on their surface. Can we use some kind of antigen to pull out the exosomes that we want and leave the others behind? Can we even tell which are the exosomes that have the rejuvenating power? And potentially more work to make this technique more efficient. The lifespan studies need replicated with different dosages, different schedules. We need to combine removing old exosomes with adding young exosomes. Nobody has done this yet, even in animals. And I have a hunch that there is a central clock in the brain. This is based on work of Claudia Cavadas and Dongsheng Tsai. Claudia is in Lisbon and Dongsheng is in New York at Columbia, I believe. They've both documented evidence that the hypothalamus has an aging clock within it. And putting these ideas together, perhaps the exosomes from the hypothalamus have some special value, and perhaps rejuvenating the hypothalamus with exosomes. Can we target exosomes through the blood-brain barrier to the hypothalamus and get a much greater effect in that way? I think it's not time yet to jump right into human trials. There's a lot of work that needs to be done on safety and in optimizing the potential for rejuvenation from exosomes. But I think it's the most promising avenue. Maybe not for dollar investments. I can't say that this is going to be a moneymaker. It certainly hasn't been so far. But if you're looking at the science rather than return on investment, I'd say there's no better place to be investing our time and our research than in translating exosome rejuvenation from rodents to humans. Thank you. I am ready for the next phase. More questions, or did
Okay.
John want to say something?
Well, that was great, Josh. Very
Outstanding.
inspiring. Yeah. And we have a few questions here, but I just want to add one comment. Turns out that as of recently, I am an investor in Mitrix, and I've attended a couple of their presentations, and they're using a bioreactor. They take the mitochondria and actually multiply them, and then they put them in what you might think of as an artificial exosome and squirt them back in. And they've had some success with animals. But then we see Albert has said something about, well, go ahead, Albert, speak up, and Steve as well. And then we got to get on to Johnny. No?
Sorry, everybody, I had trouble connecting, and my image is upside down. I'm still pretending to be a bat, so I'm just showing you that, and I'll turn it off. But in terms of what we're looking at doing with exosomes, I urge us to do the simplest things and measure basic vitals before and after any of these procedures, because I believe we're going to find a strong signal there as well if we just look for it. As if anybody's done it, please let us know what you found. I'm thinking about blood pressure responses, body temperature, and other things we can measure in real time, heart rate.
Keep in mind that Josh cannot read for some reason the chat. Maybe that's just a technique problem, but if you, Steve and John and Diane, if you have comments, please make them verbally at this time.
I just want to say I'm an OBGYN and I've been pouring plasma into people for 35 years, probably more on top of residency, more than you can imagine. And I can't remember, and I say this all the time in my presentations, we poured, you ever see a postpartum hemorrhage? Pregnant women know how to bleed really bad. So I can't remember the last time I saw a blood reaction with a placenta previa postpartum hemorrhage. So I don't know that I completely agree that it's all about exosomes, because you have to consider the extracellular matrix. You have to consider the blood supply to the organ. So I think it's more complicated than that, but I'm a little confused. If young plasma, if the kids are checked and they're between ages 18 and 24, and we know all the healthy stuff in them and they contain 37 billion exosomes, why wouldn't you take 20 cc's of young plasma and put it into people? That's what we're doing clinically. I'm a little confused about why you'd worry about pigs and this and separate that. Just take the human plasma. 20
yeah, there's an answer to that. So far, in the experiments in Nanjing and in Harold's lab, they're using massive doses of exosomes, overwhelming the number of exosomes in the old animal with many more young exosomes. So say you want 10 times the exosome in the body of an old person, you need 10 young people to treat one old person. But you can't take all of their blood. You can only take 1 pint out of 8 pints. So you need 80 young people to treat one old person. And the numbers just don't work out. You can't get 80, 18-year-old volunteers for every one person that you want to treat.
I don't know that I agree with that, because you're not taking blood, you're taking plasma, and you can donate plasma twice a week. And I'm just telling you, in 20 cc's, there's 374 billion exosomes in a 200 cc. So the volume of exosomes, I'm just saying in little baby amounts. If you want to use massive amounts, you give somebody a liter of plasma, you're giving them over 1 trillion exosomes. And again, you can collect plasma from young kids twice a week. You can collect plasma from anybody twice a week. How do you think the Red Cross does it, and all the drug companies to get all their stuff? They pay for plasma.
I can comment on that, since I'm a person in the plasma collection business. You have 5.5 trillion exosomes in the 3-liter treatments that people have been receiving. 5.5 trillion, plus all the proteins, peptides, hormones, cytokines in a complete natural balance. Now, we collect on average 750 cc's of plasma from each donation. We are unique in that we have a freezer capacity where we then collect multiple donations to get one individual who can provide 1 liter or 2 liters or 3 liters of treatment. So we really stay on top of it, but the therapeutic quantity of exosomes is massive, but it's a natural balance because it's just straight out, no concentration. But the most important thing from my perspective is that exosomes send a signal, but they have to have all the cohorts, the hormones, the proteins, the peptides, the cytokines, to be able to enact that signal. So all this talk about exosomes, especially if they've been biologically sterilized, because the other thing we know is that quantum biology requires collection and preservation and testing for its pathological pathogen purity, but you don't sterilize it, because when you do, you then end up with a binary substance. And we have physicists who've even opined on this. So really, the reason we collect, and then we immediately, after we take a sample out for testing, immediately—
Excuse me, what's a binary substance?
we have, well, I'll send you something on quantum biology, far more complex than a synthetic drug or a sterilized biologic. And I'll give you all that detail. But there is a reason that heart transplants, let's say, must be, once they've been removed and put into a cold nutrient bath, must be transplanted within 4 hours. If you want to do it 12 hours later, it's not going to revive for you. So there is an element of where you keep life preserved. And we saw that with the nematodes that they pulled out of the Siberian permafrost and revived them after 46,000 years. And they've now reproduced 100 generations perfectly. But if something dies like a woolly mammoth, you're not going to bring it back. You have to clone it to an Asian elephant to get any life. So the preservation of life, which is why when we collect the sample and take out the test, we preserve it at minus 80 degrees centigrade up until just thawing it out before somebody administers
Tom, I'm suggesting, I want to suggest you're getting off topic here.
What, excuse me?
Let's stick to the science of exosomes and not talk about woolly mammoths.
You know, actually, they're all connected. You know, so you're limiting your scope of understanding because there's a reason that, let's take IVF. IVF is at ultra cold temperatures. Once you source an egg or collect sperm, transplants we've been doing it since the 50s and 60s. The heart transplant was 64. That all requires a very narrow time frame and immediate preservation of the organ before transplantation. There's a reason everyone goes through all this trouble to preserve life rather than let it die and think it's going to live for you. Plasma has a lot of attributes that sterilized biologics and that synthetic drugs don't have because they don't communicate to our body at a quantum biologic level. I'll get you that detail, but it's not just about exosomes. There's a reason that we have all those cohorts. It's the complexity that we're underestimating, both the fact that it should be a living substance, only life begets life. Dead substances, like a dead heart, is not going to be put in your chest and is going to start beating again. So we're underestimating by trying to find a commercial simplicity of what is important to preserve and the complexity of the therapeutic we're trying to provide. You're talking about a magic molecule, exosomes. All right, well, there you go. That's one component. Oh, let's get some proteins. Oh, let's get some peptides. Oh, let's administer some cytokines. You're going to sit there with a menu that you're going to try to figure out, or you can pull it out of us when we're optimized between 18 and 25 for our biologic peak of fertility, which is our purpose in life, is to reproduce. After that, we become expendable. So you capture that plasma with all that complexity, preserve it at someone's peak of life, and infuse it into somebody old like me, and the effect is amazing. But we underestimate when we go into this magic molecule mode. Let me get you GDF11. Well, there's only 4,999 more peptides. So I guess you have GDF11, 12, 13, 14, or you can pull it out in a natural balance from a healthy individual, have it be pathogen tested, keep it alive, and put it in you. And it's simple and it's straightforward and it's less expensive. It's 1/10 the cost.
If I may intervene for a second, what I would like to do, there's several questions here, and they're all very interesting, but I think what we should do is let Johnny get in and say his piece so that it kind of, we can consolidate the questions and so on. Johnny, can you do that?
You bet I can.
Okay, there he is.
Yeah, I have a lot to rip through here in my 10 minutes of fame. Let's hold questions to the end or for another time. If we don't have time today, we can reconvene. So first, I would like to frame all this in something that should resonate with you, and I need to share the sound. Yeah. Okay, so it's like this. The rich guy's going to the grave, the Grim Reaper's standing there. See, he's pointing. The rich guy has sacks and sacks of money. Leave it all right there, buddy. Can't take it with you. And what does he say to that? Yeah, kind of like that. So here's what I'm gonna do, or die trying, but I intend to be fully successful. So it's like that. But we're fighting a battle of time. And the irony is we need more time to get more time. And that kind of reminds me, I need to turn on my timer here. So 10 minutes minus a few seconds. So, okay, using the time we have, and I'm 74 blinks, they're not years, I call them blinks because that's how fast they go by. The most effectively with the therapies are going to get us over the line. So this could be titled The 3 Most Important Therapies. So this is what I'm focusing my time on, 3 things. First of all, let's talk about me. This is my foundation. I've done a lot of stuff over the years. I've been active in various forms of investigating and using plasma since 2015. Here's a long resume. So enough of that. Thank you, Josh. That was an amazing presentation. And Walter, your efforts are really appreciated. It takes a lot more time than it looks like to put together these shindigs. So thank you. Thanks to both of you. So we'll call what I'm doing a triad, 3 things. The one in the middle is advanced exosomes for even greater system-wide age reversal. And Josh touched on some of these, and the others did too. The source of the exosomes, the type of the exosomes, what they do, and the quantity, which has come to light recently. Now, that was number 2 in my triad. Number 1, the first thing, of the various therapies that I've done, the very best, and I've done a lot of different therapies, the very best objectively and subjective measured results I've had were therapeutic plasma exchange. My expectation is that therapeutic plasma replacement will be even better. And this is something that could be done now, and it can be done now. Diane and Tom are doing this in Texas right now, and this is a bridge to the advanced exosomes that don't exist now. If you can tell me a lab I can call up to order some, and a doctor who will put some in my arm, I'm all ears. The therapeutic plasma replacement will be a bridge to creating advanced exosomes. And a group I'm working with here in Florida is working on this. We're in discussions with some people, and we're going to test it in rhesus monkeys, by the way, before we move on to, as quickly as possible and safely as possible, move on to humans. But then together in a few years, those 2 would be a bridge plan to what I believe is the ultimate Yamanaka or OSK gene therapy to reset the epigenome towards youth, also known by some as cellular reprogramming. And all the while, and I'd love to dive in with Tom Benson on his matrix, mitochondrial therapies, and senolytics and AI. I'm doing a lot with AI, but we got to focus. So I'm focusing on these 3 that I'm sharing with you today. Like I mentioned before, the best result I've had that I believe is available now is TPR. So what is it? Remove plasma from seniors, replace with thoroughly screened plasma from healthy donors in their 20s. And there's an important note. This makes for better health in both the old recipient and the young donor. And I'll explain a little bit about that a little later. Some of the benefits, well, no, I'll explain about that now. I put together a little write-up. You're welcome to have this. Here are some of the background on just how safe it is, how many apheresis, plasma draws are done a day, how many liters of infusions of plasma are done every day. Here are some write-ups from different organizations. I'd love to linger on this, but here's a search with some sources. Our dear friend Vince Giuliano has written about this. So I'm going to get back to this. So you've seen this before. I think everyone here is familiar. The concept is old mouse shared young mouse blood and vice versa. Old mouse got biologically younger according to the biomarker tests. Young mouse got older. Same principle applies to humans. Okay, TikTok, I'm going to have to accelerate now because I have a lot of material to cover. I'm going to do this when allotted more time, possibly on my own channel or my own meeting on Saturday morning sometime, but I'm going to have to accelerate. So anyway, donor's blood tested for genetics, antigens, heavy metals, APOE. Here's a raging controversy about spike protein. I don't have all the answers to this. I hear both sides, but we do have an IRB approved clinical study. I have that here somewhere. Yeah, that's what it looks like, just to show we're legit. My organization is an IRS approved 501c3 nonprofit. There are variations of TPR, TPE, out with the old, in with saline and albumin. Albumin is great stuff, and just a straight infusion. This is a comparison study where we're going to compare the results of all three. Like I mentioned before, the best objectively measured results, and I'll get into what the tests were at another time, but the best I've ever had, and I've had cord plasma infusions, exosomes, GDF11, Clotho, and I expect when I get to it with this study to get my own plasma will be even better. So plasma's the liquid part of blood. You can watch this on the replay, but here, plasma contains thousands of compounds, exosomes, EVs, acellular nanoparticles, whatever you want to call them, cytokines, growth factors, metabolites. You can read this later.
Johnny, if I may just interrupt for a second, I want it to be clear that you don't have to hurry. People can step out whenever they want.
This is so damn exciting. And, you know, we're on the verge. We see daylight for this aging stuff. We really do. Back on track here, this doesn't exactly name the types of exosomes. It's an ever-evolving naming convention, so I've read anyway. Here are a few more examples of cytokines. These are a few of them I've listed here. There are lots more. Growth factors, mitochondria. Tom Benson suggested I put this in the presentation. miRNAs. I'll suggest if there are some detrimental substances on this list that they are more diluted than are floating around in my 74-year-old body right now. So that's a good thing. Less is better than more of bad things. So goes on and on. You can read at your leisure. Here's my take-home point. This full range of balanced and complementary components found in plasma from young people would be far more effective than one molecule or even several in combination. And this balance is consistent with the target we're aiming for, youth. At least that's what I'm aiming for anyway. And there's evidence that these assist in dementia, Alzheimer's, and Parkinson's. Josh and I may disagree on this point. Disagreement is good. Let's find out. Okay, it doesn't actually look like this, but this is intended to make the point that they all interact with one another in a big complex network. Here are just a few published studies. You can have this PowerPoint. You can look these up yourself. I'm really far more interested in humans than rats, but most of these at the start anyway are humans. Then there are one or two rat studies in here. Goes on, and you can search clinical trials. You can do a PubMed search. Goes on. Some luminaries are interested in this. They've either published on it, they do webinars. Even Peter Diamandis, he's a big deal in his arena. He has TPE treatments. Here's some examples of medical conditions approved for plasma infusion. Some of the things we've learned on the studies are here. I'm not going to read these to you. You can go through the replay and read them for yourself. The take-home point to this is the current evidence demonstrates it's worthy of clinical study to evaluate aging biomarkers. And Diane and Tom are doing that in Texas, and I'm working on getting this going here in the free state of Florida. And I have other references for you. They're less formal. There's some testimonials and anecdotes. You know, if we ever talk, if you're really interested, I would share them. So, full range of balanced and complementary components consistent with youth, interact in a complex network. Now here, music lovers, one therapy is like a piccolo play. Okay, beautiful. Combinations can be like a string quartet. Lovely. Now, hey, sometimes the wrong combinations are more like a loud, out of tune, obnoxious rock band, intoxicated rock band even. You know, we go through anabolic and catabolic stages, growth and repair, and some people just throw everything at it where the therapies are fighting one another. So it'd kind of sound like that. But young plasma is like hundreds of symphonies and choruses, all in harmony. Okay, that's lovely. And there's evidence that therapy is the brain. I kind of think that's what dementia and Alzheimer's and Parkinson's sound like. So here's some of our team members. Charlie Mall, he's one of the study site physicians. Joe Parita. Debbie O'Neill has 30 years in plasma and blood banks and labs. She's outstanding. Krishna and I work together on developing that AI system to evaluate questions about aging. So he's in on this to use advanced AI to evaluate the biomarkers. Aging is 100% fatal. No way to these things. That's what I say. You can't take it with you. This guy tried. How'd that work out for him? Okay. If money's really so important to you, think of how much more you could make if you just had more time and how much more you could do. Okay, so my groups and I are working on open-ended life as long as we want to, and of course, healthy. And, what are we going to do with all that extra time? Travel? Party? Well, yeah, a little. But I just suggest that it's far more fulfilling to have a purpose, and that after we solve this aging thing to make the world a better place, here's some of the things I'm going to do and some of the people I admire. Mark your calendar, December 17th, 2049. I want you there. My big birthday party. That, my friends, is where the long-term planning will begin. We'll do things we did when we were young. Here's some of these. We'll have some laughs. Whoa, too much umbilical cord plasma. Yeah, this can be a profitable business. If you're from Sweden, that's what your money looks like. I have skin in this game, man. I've put a lot of my own money and precious time and moved to Florida to do this, into making all this a success. I'm not in it for the money. I just want to be first in line for the plasma, and to also, I moved to Florida to take part in the advanced exosome research and solutions development, and then alongside and along with the Yamanaka OSK. So yeah, I'm going to just go through this. You can read these if you want.
Yeah. Johnny, how could people get back to your source here and speak with you or access these slides?
I'm going to wrap it up, then I have a whole slide for that. I'm almost there.
Okay, thank you.
So here's what I'm going to do. And that's how well-timed. Great minds think alike, Walt. So that's how you can reach me.
That was really great. So let's see. How are we going to do this with all these questions that have accumulated? I see that Albert has had his hand up for quite a while. Let's go ahead and let him have a moment, and then we'll.
Thank you again, Josh, for that great overview. My question is about what level of plasma or exposome replacement do you think is needed in animals or humans, if you can guess, to be effective? Can we be replacing just 10 or 20% and be hopeful for good results, or is there a much greater percent you think is needed? And if others are showing success with smaller numbers, please speak up after Josh. Thanks.
As far as I know, there have only been less than 20 rats and mice who have received this treatment. Harold is really clear that the dosage is something of the order of 10 times the exosomes that are already in the rat. So overwhelming the old exosomes with young exosomes. The dosage information wasn't clear in the paper from Nanjing, but I did a back of the envelope calculation backing that out of other data that they gave, and I got the same impression that they're using overwhelming quantities of young exosomes. Whether you can get results from smaller quantities, there's a lot of research to be done, and let's get on it doing it. There are smaller dosages of plasma used in humans in plasma exchange, and as far as I know, I hear mixed results. Some people are saying that they've gotten wonderful results from plasma exchange and they rave about it. Other people say, well, I'm not sure I felt anything different at all with the same dosage, which is some percentage of your plasma being exchanged, not the whole amount. I'll let Johnny comment further.
Okay, there is evidence of this as far as plasma. A very knowledgeable person in this area indicated that he has seen that 1 liter of plasma gets good results, measurable results. 2 liters is better. 3 liters for 1 treatment is much, much better. Now, I trust my own experience, which is with TPE only. I'm looking forward to TPR, best results ever. But I have a couple of personal friends who went to Texas to have the 3-liter treatment, and they claimed, and these are people I trust, they're in my inner circle of friends.
Whose lab is this in Texas, Johnny? Who's doing this in Texas?
Diane Ginsburg, who is on this call. I think she's still here.
She had to go get patients.
Oh, okay. Well, Tom is a part of that.
And Dad Strickler is doing this too?
Yes.
Yeah.
We've filled the prescriptions, not 20 rats. We filled the prescriptions for 1,000 people. This has had far more broad usage than people really perceive. And I myself have probably received as much young plasma as anybody over the past 7 years. I've also been around TPE since the 1970s, originally as a saline supplier. There's a real lack of understanding of these treatments. Like its apheresis, like its sister technology, dialysis, is cleansing, but it's not restorative. You're not going to put saline in you and have your body get triggered. It just doesn't happen. No one performs more apheresis procedures per day than plasma collection centers. It's the same machine. So there's a very distinct difference. You see somebody can be on dialysis, but their kidneys never get better. There's an individual here in San Diego who has had total plasma, 3-liter total plasma exchanges more than 1,000 times. The other point is apparently he's not getting better. So it'll clean you, but it won't restore you. You've got to put in those youth factors. That's why it's so complex. And that's why those youth factors, I was a sterile saline supplier. There's no life in sterile saline. You've got to do biopreserved plasma. But we've done 1,000 prescriptions we've filled. We use 10,000 units of plasma a day in this country. It's been legal since 1938, and since 1942, 20% of the entire blood supply has been from young donors. So we have far more experience in this than, and comparing TPE also to plasma treatments, than most people really understand. or have any appreciation for.
Wanted to point out that we've had many people receive partial plasma exchange, and nobody has turned from a 70-year-old person into a 30-year-old person yet. The results don't compare to what we've seen in rats, where they look and act like much younger rats.
Well, again, 30,000 TPE procedures are performed in the United States in major medical centers per year, 30,000. And it's been going on since the 1970s. Again, I was a saline supplier. And 150,000 plasma donations using an apheresis device, replacing the removed plasma with saline, are performed every day. No, it doesn't bring you back. It cleans you. It's very, very beneficial, especially if you're a young donor and you donate plasma frequently. You can reduce your inflammation and maybe you won't get diabetes. But it's not going to take an older person and regenerate them. It's just not. You need all of those cohorts. You need the replacement, and saline is not a replacement for regeneration.
At one of our meetings, one of our Saturday meetings, Diane once framed it in two ways. One was that as we get older, our bodies no longer have the capacity to rebuild components in our body like we did when we were young. And she kind of drew it home where a TPE is like an oil change, and it's great. I've experienced the benefits, but TPR is like a partial rebuilding of the engine. How much? Well, I have the conviction that it's going to be a lot. But remember, for me, I'm all on board with exosomes, and exosomes is the second part of my three-part focus right now, which is the theme of my talk.
Again, be careful about exosomes if they're sterilized, because you're not talking about the same thing in commercial products to have that storage, shipment.
I see there's several things up on the board. Oh, here's come somebody. A nice looking dog. Tom, we should not assume, in fact, it's almost certain not to be true, that even the youngest human actually has an ideal ratio of everything. That's simply false. Nature never delivers perfection. It always delivers a compromise.
All the donors are extensively health screened, and we take protein tests, assess their protein before every donation. So it's probably about as good as you're going to get. Certainly there are variances, but we only collect from very healthy young college students, and they are excruciatingly medically examined before every donation.
Well, I hope I didn't either express or imply that young plasma was perfect, but it's a first step in my personal bridge plan on to developing the others, and then building a better world.
Having received young plasma, a couple things I can tell you is that if you get 1 liter, it's beneficial to you, and I've seen that in some of the testing I've taken post infusion, but you don't really feel it. You get 2 liters, you feel it. You get 3 liters, and you have to hold yourself down on the ground. It really powers you up. For probably since 2018, I've been getting roughly 2 liters a year. Then when Diane's study went to 3 liters, the last 2 treatments I got per year were 3 liters. Huge, huge difference. It is volume related completely. But the one thing that constantly gnaws on me is I've been doing it once a year, and my body, boy, 6 months in, is saying, bring back the juice. It is very, very noticeable at the higher volumes and beneficial at lower volumes, but not as appreciably noticeable.
Well,
what should we do, folks?
John made a comment that he personally doesn't think a priority is plasma transfer. I would welcome a private conversation with you or now for reasons why that is so. And let me just, my endpoint is open-ended healthy life. So, and I can shift my priorities on a dime, and I have some resources.
I don't mind.
Whenever whenever you'd like to have that conversation, now or later, or I'd love it.
Go ahead, John.
The reason is
Let me
Yeah,
summarize
well-
where we are when I hear some people saying that exosomes aren't enough. We need other ingredients in the plasma. I hear people saying, well, exosomes are more than what we need. There are specific components of the exosomes, maybe just the mitochondrial, maybe some RNA components. And these are things that urgently need to be tested. These are experimental questions.
Yeah.
These are not questions for philosophers. Let's get into the lab and find out.
Agreed.
Why the lab? Let's just do it in a clinical setting with humans, and there you go. This whole indirectness of labs and rats and everything else has its place, but that ended at about 2014. And when Stanford transitioned over to their AD and PD studies with humans, and it was only after Grifols bought it that it then went back to a sterilized, source plasma product. But humans are the best study subject. Plasma has been legal since 1938. And there you go. That's about as direct as you can get.
Good point.
If I could make a quick point, I am a DIY self-educated biohacker, and I absolutely believe and support the idea that young blood generates greater feeling of younging and a younger gene expression. But doing plasmapheresis on a regular basis for the rest of my life, I don't see that is a sustainable process. Am I going to be 85 and I'm going to fly to Honduras to get it done in an affordable clinic? That's what I think about. And that's why I think this Chen et al. 2024 study is so critical, because they posit that the regenerative effect is partially driven by small extracellular vesicles via increased PGC1A expression. Now, I hear you, Josh. We don't know to the degree to which it's true. I don't think they're arguing that's the only molecular target of these exosomes. They appear to be saying, as I read it, that it's the major one. And there is a coincidence of other youth and health promoting interventions, hyperbaric oxygen, PEMF, heat shock, cold shock, that also increase PGC1A. So the coincidence of this focus by younging techniques on PGC1A is certainly intriguing to me, because when I'm 90, I'm not going to be flying to Honduras to get plasmapheresis done, and I don't imagine it will ever be affordable in the US for me.
Yes, it is. And you got to be out of your mind to go to Costa Rica and spend $15,000 for TPE, or even go to Kiprov and spend $10,000 for it, when you can go to Grifols, let's say BioLife, Takata, all of them, and it's a smaller volume, but they'll pay you to undergo the apheresis procedure. So you can either go donate plasma and get paid once a week to do that dilution with saline, or save your money and go to Costa Rica and spend $15,000 with the same machine, because if you do 3 plasma donations, you've achieved fundamentally the same volume of replacement and removal.
Well, Tom, a really good point raised here, of course, is the accessibility to the masses of this thing. Are we going to something that will only be available? Yeah.
There are 1,000 PPTA, it's called, Plasma Protein Therapeutics Association, which is CSL, the largest, Takata, Grifols, all of them. There are 1,000 collection centers in the United States where they will pay you basically $35, $40. We reward our students $100. We have no problem getting donors, but they're getting that treatment. So you go in 3 times during a month, Yeah, you made yourself, lunch money, but you got the same therapeutic benefit as go visiting Dobry Kiprov. So it just makes no sense. And if you take blood banks are not as interested in plasma, but if you combine them all, there are a couple thousand places in this country where they'll either do it for free or they'll pay you to donate your plasma using the same apheresis machine. So you got to be out of your mind.
We're talking about donating 3 times a month. We know that some of the components of plasma are replenished by the body faster than others. How long before you
48
have
hours.
the full? I haven't asked the question yet. What is the evidence that the exosomes come back in a young person in 48 hours?
Because that's the legal foundation of collecting plasma, and that's been since the 1970s. It's legal in the United States to actually donate plasma twice a week. We don't at Spectrum do that. We'll do a maximum of once a week, and everybody before they donate.
Yeah, I mean, I'm more interested in the
protein test.
experimental measurement than in what the law allows.
Yeah. So if I could just speak to,
We do it
I have done a lot of
150,000 times a day, and you're waiting
Yeah.
for a couple rats in a cage to give you information 150,000 times a day. And we've been doing it for decades. We actually figured some of this out.
I'm completely on board with everything you've said, Tom. I've investigated this in detail. I've spent a couple hundred hours looking at it. I'm about to get my 50-gallon badge at the Red Cross for my blood donations every 8 weeks. The problem with Grifols and I've looked into that in detail. They have an age limitation. There's an age limitation on whether you can donate, and also so that there's a geography limitation. Because they don't want to have people donating too often, you have to demonstrate residence at a certain town with an invoice from the phone company or a water company or whatever. And I'm in a locale where there is no Grifols blood donation. My wife is a nurse. I looked at eBay to buy a used plasmapheresis machine, and there was one on eBay for $2,000. I was ready to pay the money. Problem is, the company that manufactures that used device, it's no longer their prime manufactured product, and they don't support it with parts and labor anymore. And these other devices are 20 to 40,000. I'm into DIY control of my fate, right? I completely support everything you've said. I believe it, but I'm looking for something that works for me. I've gotten pretty radical, and I want you folks to straighten me out about what I'm missing. I'm not a professional. I'm a fucking software engineer, right? So I make it up as I go. Here's my thought. I've been donating at the Red Cross. Plasma replaces itself within 10 days, right? Platelets.
Yeah. Blood takes 2 months.
Within 2 weeks, and white blood cells within less than a week. The only reason for the 8-week period that you have to wait for Red Cross blood donations, as I understand it, correct me if I'm wrong, is you're waiting for the red blood cell volume to increase in the blood circulation. Turns out that there are now home kit hemoglobin tests you can buy on Amazon for $100, along with some sticks to do testing like you can test for ketogenesis. I bought those. I did a blood test at the Red Cross, have the home test kit. I get comparable number results, the hemoglobin cross on the Amazon home test kit. So I'm thinking, my wife is a nurse, I'm thinking I want to donate more often, complete whole blood, more often than every 8 weeks. What do you think?
No, I'm a fully accredited blood bank.
Okay.
And you're correct, it's the removal of the cells that is debilitating to our body. The upside is, especially if you're a European male, you lose some iron. We don't have the benefit of menstruation to kind of protect us so it can accumulate, but it's fundamentally debilitating. Plasma is not. Plasma's rejuvenating and cleansing to a certain extent. So there's a huge difference. And the trouble with blood donations, for the benefits that you do receive, because you are pulling out plasma, of course, is that you can't do it frequently enough.
Right.
So you're just not going to get there. It's important, but it's all about the plasma. It really is. And the plasma you can remove frequently, and that gives you the greatest benefit. Blood just won't be able to do it. You can't do it enough.
Isn't there also a finite number of times? That is to say, you only have a finite amount of blood production in a lifetime. That's a problem. If you keep donating, I would think you're basically forcing your bone marrow to produce more red blood cells. This may have consequences. Am I wrong on that?
And also, depleting your white blood cells can deplete your immune system. I donated at Grifols for 5 years until I turned 70, and they stopped allowing me to donate. I know BioLife has an age limit of 99 years, but you have to be within 40 miles of them, and I'm not. So I haven't been able to donate for a couple of years, but I sure would like to donate plasma, but I would not ever want to donate blood.
I would agree, and there are a lot of members of the PPTA, and they all have different criteria. So you may find Takata will take you where Grifols won't, and CSL, the biggest, may take you. Yeah, it gets to location, but if you're fortunate enough to have the location and one turns you down, you may find another one that will accept you. But blood donations are not a viable rejuvenation therapy.
I agree.
BioLife, if you're over 70, BioLife may take you. You have to go through
Yes,
some
but they're more than 40 miles away from me.
Yeah, you want to hear something really scary is, where we take out the plasma and we biopreserve it at minus 80 degrees centigrade, all that stuff. Grifols or Takata, CSL, once they collect the plasma, they then put it in ethanol for 7 to 9 months to separate out that plasma protein. That is definitely a dead substance. And not only that, but that 7 to 9 month manufacturing cycle, which again starts with a donation, then makes it 10 times more expensive than just getting a good donor and receiving that plasma directly. So there was a time when the blood supply was unsafe, and Grifols and that ethanol was a great salvation. But that ended in the year 2000 when pathogen testing got so sophisticated, you didn't need to sterilize the plasma anymore. And so you have an old industry that represents 20% of our pharmaceutical supply controlling and hanging on. And yet for the past 24 years, they are fundamentally biologically irrelevant because there's no need to sterilize the plasma and destroy all that bioactivity at that cost. It's kind of like going to Costa Rica and spending $15,000 for something somebody will pay you, rather than taking the donor direct arm to arm. You send it off to Switzerland for 9 months to swim in ethanol. It makes absolutely no sense in the current context of the last 24 years.
I appreciate the feedback very much.
Well, we've been going for 2 hours. This is fascinating stuff. I think we're very much at the cutting edge here. But should I put an end to it, or do we want to keep going?
Walt, if I may ask one last question, which I put in the chat, but we skipped over. Back a ways, I asked about the idea of whether or not we meaningfully have, or any mammal has a single biological age, given that all our organs and tissues are turning over at different rates. And if you just compare, teeth and skin and hair and nails, blood, a wide range of rates. And is there anybody who's come up with a way to integrate from our youngest age, whatever that might be, to our oldest, to see which is driving us faster towards death?
You've got a good point. Different tissues in the body age at different rates, and there is some consensus around the body. And I'm betting on the idea that that consensus is established through exchange of exosomes.
Geez, Josh, if it all was about exosomes, they would be more predominant, I think. I think it is all the ingredients. It is the peptides, it is the cytokines, it is the hormones. It's it all. So this selectivity of here's the magic bullet underestimates the complexity of
Tom,
regeneration.
I'm willing to consider that. I think there's a lot more research to be done. And just remember that there was that one small study. It's certainly not the definitive be all and end all, but there was the Smith study that looked at rejuvenation with exosomes alone and with plasma, no having the exosomes removed on the other hand, and they found no benefit from the plasma without the exosomes. I'm not saying that's the definitive study. It's one data point. We need to do more research.
Yeah, but that's like having a radio and not sending it a signal. So yes, you need the exosomes because you need that signaling, but you need to respond to the signal. So taking out the exosomes removes that signaling factor. So it makes absolutely no sense, which is why I think the decomposition of intact organic natural plasma from a young donor makes absolutely no sense, because you're deconstructing what nature has perfected at our peak of biologic life. It's pretty much that simple. Nobody wants my plasma. And like some of us old people have been saying, even Griffles, when they throw it in ethanol, doesn't want it. It is all about the young. That is our peak. It's not about cord blood, because you're not going to find that plasma in that environment is sustainable for an adult. We're taking adults at the peak of their life, so they have hormones and everything. I don't collect from anybody who's younger than 18, because I don't want to have some male come in there who still hasn't got his hormones straightened out. And you're certainly not going to find that in cord blood. It really is almost very simple. Just capture the plasma at the peak of performance, preserve it, and put it in you, and you got it all. And there's no mystery. Nothing's missing, and it's all in balance.
Maybe you're right. Let's do the experiments and find out.
I see that John Hemming is disputing some of the things here.
I have a view, but it's not one to necessarily spend time on today.
There's one more thing, get out quickly, that should be mentioned in this context, because we're talking about the blood. Mother Nature established a mechanism in mammals, including in humans, called the cholinergic anti-inflammatory pathway. It is a mechanism connecting the brain to the spleen through the vagus nerve system. And hundreds of scientists in hundreds of studies have documented the fact of the existence of this mechanism. The larger mechanism is called the inflammatory reflex. The anti-inflammatory mechanism, brain to spleen, that reduces TNF and other inflammatory cytokines in the blood circulation is called the cholinergic anti-inflammatory pathway. It's been demonstrated conclusively that triggering the CAP, the cholinergic anti-inflammatory pathway, increases survival probability in rodents and in humans. So that's another blood sort of daily intervention that can be done that I happen to believe impacts the same blood factors that both younging factors and plasma dilution addresses.
John Hemming, did you have something else you wanted to include? It
Well,
seems like you have very, very well-informed opinions.
Well, whether I'm well informed is a question that only time will tell. I'm very happy. Yeah, it's one of these things. I'm very happy to discuss my own opinions. But we've got to the end of this, and everyone's like, yeah, we're at the edge of where people are thinking of departing and getting on with other things. And to be fair, I have other things to do as well. Exosomes are important. Macrophages are important, but at the core of it, in my opinion, is the efficiency or the mitochondrial membrane potential and the burden of senescent cells, both of which have an effect on gene expression. And you don't have to agree with me. I have a reasonably well-held view, which seems to fit with experimental work as well, which is biohacking experimentation.
John,
But
I'm eager
I
to,
do have
eager to learn from you, and I will read everything you send me. And the idea that mitochondria have an effect on reprogramming the nuclear epigenome is
Well, not
completely new to me. And I'm
reprogramming. Reprogramming is the wrong thing. It's very obvious, though, is that the level of nucleosolic or cytosolic acetyl-CoA drives splicing variations. It's just not really news. And obviously, the level of acetyl-CoA in the cytosol is driven in part by the amount of citrate that comes, only in part, and that's important, by the amount of citrate that comes out of the mitochondria. So the mitochondria, I don't think that the idea that the mitochondria have no effect on gene expression has any support at all. But, you know, I'm happy. Yeah. If I were to be presenting my own views, I would not drink anything. I'm quite drunk at the moment. So, you know, it's probably not the best time for me to explain things.
It comes off better than drunk Shakespeare, so congratulations, John.
You know, I'm looking at Tom Casey, and he has a remarkably good complexion. I think he's as older as me, and he's probably had more of this kind of therapy than any of us individually, based on what he said here today. But yet I do notice that his hair is still white.
Yep.
So there's maybe a limit to how far back we can go.
I have an interesting hair from earlier today, which has turned from white to dark, which is curious. I could try and find it somewhere, but still.
I have a question, please, for everybody who's looking at the effects of treatments meant to be anti-aging. Would you expect people who are responding to show slower or faster growth of hair, nails, anything else they might be measuring once they are successfully younging? Does younging mean we grow more slowly, or does younging mean we're healthier and we grow and replace these tissues more quickly? I'm not asking with any bias. I really have no idea, and I'm curious what you guys think.
I'd like to talk to that.
That's a turtle
I don't think
question.
anybody knows the answer to that question, and I'm eager to find out.
I've gotten a lot of plasma, so I'd like to talk to it a little bit. It was Jesse Carmasian with ambrosia that started saying people's hair was going to come back. And there's organizations that think that they're going to, you know, push you back in time. I think really what happens is you make yourself your best version of who you are today, but you're not going to go back and turn back the years. I physically look better than I did 10 years ago before I started receiving the plasma. And so, you know, your skin, everything, which is kind of the amazing part, it's a systemic treatment. It's not targeted. You want to give it to somebody for Parkinson's and they find out that their eyesight and, you know, their physical capacity, their libido all improves. So the plasma just goes in and addresses every single cell to the extent of the volume that's administered. So it's volume and frequency related, but it makes you a hell of a lot more energetic, you know, alert, you know, stronger, all those kinds of things, but it's not going to turn back the clock. It'll make you today as good as you can be. And I think that's huge, but I've received a lot of it and it makes a hell of a difference.
I get the picture that you're painting, but please answer if you can give us a sense of it. Are you cutting your nails and your hair more frequently with this treatment than before? Do they grow back faster or slower after you cut them nowadays, do you think?
Well, you know, I also take some supplements and things like that. So I would say I don't find it, you know, it all grows fine. I don't find it necessarily noticeable. What you do notice is your alertness, your physical energy, your sleep patterns, libido, all those kind of things immediately respond. And that's kind of the cool thing. It's a trigger. So when you put it in there, it's like when people used to get stem cells, they'd say, oh, come back in 7 to 9 months and we'll see how you're doing. Well, plasma is a trigger. So it immediately triggers you as soon as it goes in. Harvard last year said biology is 99% software, it's the messaging, and that the body is the hardware that responds, change the signal. It's like I've been trying to find a young person who wants my plasma so we can do kind of a placebo control, and I haven't found anybody. So it's a pretty straightforward opportunity we all have, and I recommend everybody get as much as you can, and as much as you can afford, as frequently as you can, is what my body tells me.
I like
What's
everything
the
Tom
cost
said.
of 3 pints of plasma?
Plasma is it's $5,000 a liter.
So if I fly to Texas tomorrow, it's $15,000 for a treatment.
Correct. But be aware, I'm a blood bank. So this is a very slim profit margin. We spend a couple $100 per unit on just pathogen testing and everything. So, there's a lot that goes into it. So I'm not quite at the, pharmaceutical level of profitability. So that's the beauty of blood banks. It's as straightforward and as affordable as it can be, but you are getting 3 liters. So you're getting a lot of volume. That's a lot of donations, and it's not inexpensive. I've tried since 2021 to get it paid for by Medicare. Medicare is all on board. It's the FDA that is stopping this, not because it's illegal, not because it's beneficial, but because their benefactors, Grifols and Takata and everybody else, this will wipe them out.
Can Johnny get his complete plasma replacement for $40,000?
When you say complete plasma replacement, what do you mean?
8 pints.
Well, it's $5,000 a liter. So
a liter is 2 pints. So we have 4 liters.
Pints.
The body is 8 pints, 4 liters. Is that right?
We've got about 6 liters in us in total on average. About 55% of that is plasma. So, I mean, we're finding the 3-liter treatment, you've got to do it over 2 days because we can generally withstand 1 liter, IV spas, everything. You can take 1 liter. If our treatment now is 2 liters the first day, but you've got to remove some blood or plasma first to make space so you don't have circulatory overload. And then you come back the next day with no need for removal, because plasma is 92% water, so just urinate over the night. And then you go in the next morning and get your 3rd liter. So within 2 days, you get 3 liters and you walk out the door. And it's an IV, so it's incredibly simple. All you have to do is take Benadryl and Prilosec just to lower your histamine response. And there you go. It's as simple a treatment as possible. We started out removing that space for TACO, transfusion-associated circulatory overload, with apheresis devices. But then we found, hey, just do a blood draw. Just pull some blood out. It's a 1/10 the time, a 1/10 the cost, and you made the volume you need, because once you put the plasma in you, you do not want to remove blood or undergo any apheresis or remove any plasma for 4 months. Get the plasma in. So taking out blood is as viable an alternative to make space as taking out plasma. And so it's an incredibly, once you hook up the IV, first you pull out some blood, then you hook up the young plasma and you let it flow in.
Tom, have you tried concentrating the young plasma just by removing some of the water?
No, I mean, I can't alter it because I'm a blood bank. And as soon as you start doing that, I don't know what the purpose is. Certainly volume overload, it may help you a little bit, but we don't mess with it. And it's all straightforward. So we're finding that a little bit of removal really makes 2 liters very comfortable. Originally, even Ambrosia was administering 2 liters in 1 shot, which I thought was inadvisable. And I started some of the largest IV companies in this country, but we found 2 liters is, and I've had 2 liters in 1 shot with no removal, but I'm a big guy. I wouldn't recommend it. And we find just a simple blood loss and getting that 2 liters in one day is very comfortable. And if you can afford it and you got the extra day, get the 3rd liter. It's an immediate trigger. You'll notice it immediately. And then after 5-10 days, overwhelmingly, you will notice it. So it's impressive to receive.
What's your address?
To speak to Albert's question and also draw another link about how important I think this PGCA 1A question is. I have an EWAT machine and I have, non-trivial cardiovascular disease. And when I am regularly on my EWAT machine for 20 minutes, the effect is like magic. I feel so dramatically better within a few days. I cannot tell you. I take it, this is not demonstrated in the literature at all, but I take it that EWAT is a form of hyperbaric-like oxygen therapy. And a reason why I like it so much is, first of all, I have felt the effects, profound effects. And the second thing is, it's a whole body remedy. It's a whole body intervention, just like the intervention to the blood circulation system. You get younging factor blood, you donate blood, you do plasmapheresis, you're impacting the whole body. So does hyperbaric and oxygen therapy, and so does EWAT. And that's why I am a big fan of EWAT.
EWAT being exercise with oxygen therapy.
So how does it work? What is exercise?
So I have an oxygen. I have an oxygen concentrator, and I have a 900-liter bag hanging on my wall. And I have a 5-liter concentrator per minute, and I fill it takes me 2 to 3 hours to fill this big medicinal material bag full of oxygen. I have a mask. Then coming, there is a nozzle on the bag that I can hook up a mask to. I have a Bowflex aerobic machine right next to the bag, and you put on the mask and you do aerobic exercise while you're breathing this highly concentrated air. And I estimate it's between 92 and 95% oxygen. And you feel better after. And I take Albert's point that, and I understand the whole hypoxia, hyperoxia thing. I think Fraudy in Israel, his article called The Hyperoxic Hypoxic Paradox, explains what's happening in a profound way. But I just want to make a pitch for EWAT is such a wonderful intervention, and it's a whole body intervention.
I suppose that would even be, shall we say, something that has commercial potential, because you have rigged this system that could perhaps be manufactured. That's kind of a small thing, but
it already is. By Mike White of
okay.
He calls his company I think Breathing, and this is now available as ridiculously small and ineffective version of not enough oxygen to make a difference, but a great way to make some money. These used to be sold long ago at REI for people in outdoor sports, and the FDA lets them get away with it because they consider not enough oxygen to hurt you.
Is that the things I'm showing on the screen now?
Yeah.
That
my
answers.
question back to Steve is, when this is working for you, Steve, and you feel that EWAT and the other therapies you do are younging you, do your hair or nails grow faster or slower than normal? And
I forgot to mention this, Albert. I wanted to tell you, my nails get thicker, my hair grows faster, my libido is greater. And these are felt experiences, right? So this is why I am so enthusiastic about EWAT and promote it. The guy who invented this whole mechanism is Manfred von Ardenne. He was German. He was like an Elon Musk of his day, born 1907, died 1997. He held 600 patents of electronic devices, And in 1960, he decided he met Hitler and gave Hitler a demonstration of his TV set, and he won 2 Stalin prizes for helping the Soviets build their atomic bomb. He went back to East Germany, 1960. He was working, he met the guy who discovered oxphos, I forget his name. And then also the guy who discovered ATP. He devotes his book to him. He invented the whole oxygen multi-step therapy approach. It's a fantastic, incredible book. The only piece of the puzzle he didn't identify himself was creating a very, very large bag to breathe out of. The value of oxygen, EWAT, over those kind of bottles is that while you're doing aerobic exercise, your heart rate increases and the partial pressure in the arterial and venous system increases, and you're actually, it's been done, you are opening up capillary structures, right? Because of the pressure of the heart rate and the increased oxygen. And I believe what I just said is true. I couldn't point you to a study immediately, like many times I can. I can validate that statement. But the physical effects are absolutely felt. There's a ton of videos, YouTube videos on this topic.
I'm going to need more haircuts, got to clip my nails more often. I'm going to be hornier. Do I want this?
Yeah. I'm not kidding. My wife notices when, when I'm not getting enough EWAT in.
Okay, folks, it has been 2.5 hours. Wow. So should I let it go here? Anybody have something they want to say in closing?
Well, I'd like to thank you, Walt, for pulling this off, and Josh and Johnny for commenting. It's been great, and I hope you have a successful run with these. Thanks for leaving the window open so nobody had to feel rushed.
Stephen Miller and I are co-hosts for this, but this week it's me. Next month, I got to figure out something good to do for next month. It's going to be hard to top this one. Thanks, everybody.